Hello everyone. Today my research title is Exogenous abscisic acid repress rice flowing for SPK8, ABF1, and HD1 and D2 pathway. Okay, now it's working now. Flowering is an important process for plants to transition from vegetative growth to reproductive growth. And the transformation of floral origins is the result of the combined action of internal genes and the external environment. For rice, flowering is one of the most important economical traits, and the length of flowering directly reflects the length of the growth period. And the appropriate growth period is important for maintaining a high and stable rice yield. The regulatory network of rice flowering is complicated, including numerous genes and many modifications, such as ubiquitin, phosphorylation, methylation, and among these factors, HD1 and HD2 are two key transcription factors. Abscisic acid, a phytohormone, has been previously reported to suppress the plant flowering. For example, in abscisic, ABI4 and ABI5, two key factors involved in AB signal, have been reported to repress the flowering. And let me shortly introduce the AB signaling pathway. On the non-stress condition, the phosphase PP2C inhibits the protein kinase SNK2 type autophosphorylation. Then this kinase cannot phosphorate ABF type transcription factors, and finally inhibits the stress tolerance genes expression. However, when under dry condition and or other adversity, plants can produce AB hormone, and AB receptor can combine with AB, then they recruit PP2C to form a complex and inhibit this phosphase activity, and then the SNRK2 could autophosphorylation and active the ABF transcription factors activity by phosphorylation, and finally active the downstream stress tolerance genes expression. Among these ABF transcription factors, many members belong to BZIP type transcription factors, and they are involved in adversity, stress, and growth stages, including flowering. And another important family is polycomb group of proteins. They are a group of transcription repressors that target target genes through chromatin modification. And they can be divided into two parts. One is PRC1 complex, and the other is a PRC2 complex. In rice, the complex, the PRC2 complex consists of several members, including SET1, FAIR1, and FAIR2, EMF2A, and EMF2B. And the PRC2 its work is based majorly on the H3K27 methylation to repress the downstream genes expression. And this complex is very important for in regard flowering. For example, in rice, it has been reported that one of the PRC2 member, EMF2B, it could be combined with VIL2, then repress downstream genes LFI1 expression, and finally induced flowering. So here comes question. Since ABA and PRC2 complex all both play an important role in flowering, but the crosstalk between these two pathways remains largely unknown. In your lab, we previously have found that a BZIP type transcription factor ABF1, it was high induced by ABA treatment, and it is highly expressed in fatty tissues such as root, shears, and leaf. However, it is low expressed in panicle and development seeds. These results suggesting that this ABF1 maybe play negative role in plant in especially rice reproductive development transition. And the subcell location experiment shows that ABF1 acts as the transcription factor is mainly expressed in nuclear. And then we found we generate ABF1 and its homolog BZ14 double mutants, and found that this mutant is showed earlier flowering compared with the wild type, whether on long day condition or short day condition. However, this way ABF1 overexpression length showed a later flowering phenotype, and application of exogenous ABA treatment can enhance such phenotype. In line with this, we also generate ABF1 RNA interfering length, and they also showed early flowing compared with the wild type. Next, we further found SCP8.
Hallo.
Hello?
Ja, mit dem Vortrag.
Can you hear me?
Ja, du hast gerade mit dem Vortrag angerufen.
Okay. Next, we found a kinase, SN2 family kinase. SCP8 could interact with ABF1 through East2 hybridization, IFC, and in vitro pull-down assay. And we also performed in vivo phosphorylation assay and found SCP8 indeed phosphorate ABF1. We can see when we put these two proteins together, we can detect phosphorylation band. However, when we treated CIP phosphorus enzyme treatment, this phosphorylation band disappeared. And the in vivo phosphorylation assay also showed ABF1's phosphorylation band is enhanced in SCP8 overexpression length. When we use AB treatment, the phosphorylation intensity is enhanced. And we also further generate SCP8 overexpression length, and found is showed later flowering phenotype compared with wild type, and also application of ABA treatment can enhance this phenotype whether on long day or short day condition. Next, we detect the SCP8 expression level and found It is
Okay.
induced by
یعنی
ABA treatment.
کلوچه کنشا خوفه.
And we use gas staining assay. We can detect its expression majorly in root hair, leaf, seed. And very interesting, we found when we apply the AB treatment, this gene can significantly induce in root, stem, and leaf. The intensity is enhanced gradually. So we HD1 and HD2 are two positive rice flowering regulator genes. We found these two genes are significantly induced in ABF1 and BCF14 double mutants during the development stage. And in the ABF1 overexpression length, these two genes hardly expression level hardly detected. And the application of ABA suppress ABF1 express these two genes expression in all the different genetic background. And we also detect this two genes expression level in these materials during the time pounds of day, and it shows a similar tendency. Shows a similar tendency. So here, based on the above results, we hypothesize that whether ABF1 direct binds to HD1 and HD2. Use the qPCR assay, we found the region of region 5 of HD1 promoter and the region 4 of HD2 promoter. It can enrich the much more amount of ABF1 compared with the wild type. While in the double mutant, this increasement of ABF1 is significantly reduced compared with the wild type. And the use EMSR assay will also confirm the direct binding between ABF1 and with HD1 and HD2.
Okay.
And we also we further determine the special binding site of HD1 with the ABF1. We can see when we mutant the binding sites, the small letter, the binding sites disappeared. So it is the special binding site. Then we know ABF1 is phosphorylated by SP10. So we further to detect the phosphorylation effect on the ABF1 binding to HD1 and HD2. Interestingly, we found that the phosphorylation bands, the binding intensity is significantly higher than no phosphorylation bands, whether either HD1 or HD2. Now ABF1 direct bind HD1 and HD2 promoters. We use luciferase transient transcription activity assay to detect found that ABF1 significantly repressed the expression level of HD1 and HD2. However, when applied ABA, this suppression level was enhanced. Another, we also add SP10 and ABF1 together with HD1 and HD2. We found that when add SP8 together, the suppression level also was enhanced. When next we search for more genetic evidence. When we overexpressed HD1 in ABF1 overexpression length, we found HD1 overexpression could rescue ABF1 later flowing phenotype. We can see when the later it can flow in the similar as the Y type. So we next to hypothesize that whether ABF1 can recruit PRC2 complex to work together to regard rice flowing. So we use the EAST2 hybridization biopsy, and in vitro pull down assay, luckily we found this transcription factor can bind to FIE2, a member of the PRC2 complex member. We can see in the region 5 of HD1 promoter and the region we can find a high enrichment of H3K27 methylation level and Phi2 level in this region. It suggests that ABF1 can recruit FIE2 to deposit H3K27 methylation on HD1 promoter to suppress gene expression. And in the region 4 of HD2 promoter, it showed a similar tendency. So how about the Phi2 phenotype? We next generate Phi2 mutant and found that it showed a significantly earlier flowing phenotype, similar as the AB4 B14 double mutants. And appression of ABA can significantly delete Phi2 flowing, so less severe level than the Y type. Additionally, the ABF1 expression level is significantly downregarded in Phi2 mutant compared with Y type. We can see the mutant also showed severely agronomic trace defeat. So we finally last to explore the AB low in the deposition of ABF1 Phi2 as at H3K27 methylation level on HD1 and HD2. Triple qPCR was performed on the Y-type seedlings without treat and with ABA application treatment. Interestingly, we found ABA treatment can significantly enrich the HD1. Significantly enrich the ABF1 H3K27 methylation and Phi2 in the region 5 of HD1 promoter. And it is for HD2 promoter in the region 4 region of this promoter, we can see the similar tendency. ABA treatment also can enrich the high level of ABF1, H3K27 methylation, and Phi2 compared with the Y type. So, based on the above results, we drove the SPK8 ABF1 and HD1 as HD2 pathway for OAB-mediated flowing repression. That is, exogenous ABA treatment. This signal can be transmitted by SPK10 protein kinase, and then SPK8 kinase can phosphate ABF1. Then ABF1 can direct binding to the promoter of the rice positive flowing regulators HD1 and HD2. And Yifeng Wang can recreate Fi2, a member of PRC2 complex, to deposit H3K27 methylation suppressive modification on this 2 genes promoter, and finally delay rice flowing. This work was published in Journal of Advanced Research last year. And this work is supervised by Professor Zhang Jian. And this work is majorly done by a PhD student, Tang Liqun. And we also think this work was supported by National Natural Science Foundation of China and Chinese Academy of Artificial Science. And also we thank the Zhejiang Provincial Natural Science Foundation. Thanks for your attention. Thank you.